There's a new form of medicine called antisense oligonucleotides that are being used to treat neurological diseases. You will also hear these called ASOs. Let's take a look at how they work. DNA is made up of 4 nucleotide building blocks: adenine, cytosine, thymine, and guanine. They are arranged in long strands to create a message that encodes the proteins making up your body. DNA is made up of 2 of these strands. A's will bind with T's and C's will bind with G's. When it is time for the cell to make a protein, the code for the protein written in the DNA will be copied into another molecule known as messenger RNA, or mRNA. It then takes the instructions to a protein production factory called a ribosome. However, if the DNA has a mutation, the mRNA will also have the mutation, and it will tell the ribosome to make a different and wrong protein. Sometimes these mutated proteins can be a big problem and cause disease as they accumulate in the cell. If we can stop the faulty message from getting to a ribosome, then the disease-causing protein will never be made. Since the bad mRNA is only a single strand of nucleotides, a specially created single-stranded ASO can come along and bind to it. This ASO needs to have the exact opposite nucleotides of the mRNA in order to successfully bind. With only 4 nucleotides to choose from, it might seem like an ASO would bind to both good and bad mRNAs, not just the ones that cause disease. But with a string of just 20 nucleotides, there are actually over 1 trillion possible combinations. So we can make a short string of nucleotides that is highly specific to the faulty mRNA. By designing an ASO to look like a single strand of DNA, we can activate a protein called RNase H. RNase H recognizes when DNA is bound to RNA and destroys the bound mRNA. RNase H does not destroy the ASO, so it can actually go off and bind to another bad mRNA. This prevents bad proteins that cause disease from being made.